Well, hey, everybody, this is Robert, and welcome to Outbreak News TV. Now, Chagas disease is a dangerous tropical illness caused by a single-celled parasite known as Trypanosoma cruzi. In most cases, if not treated immediately, the infection becomes chronic. The immune system of the host greatly reduces the number of parasites present in the body, but it fails to fully eradicate them. And current diagnostic approaches often fail to detect these very low numbers of parasites. Now, our research team from the University of Georgia and others aim to develop a test for cure. So joining me today is Rick Tarleton, PhD. Dr. Tarleton is a Regents professor at the University of Georgia and a distinguished professor in biological sciences in the Department of Cellular Biology. Dr. Tarleton, welcome back to the show, sir.
Thanks, Robert. Good to be here.
Excellent. We've talked about Chagas on this program in the past, but for people who haven't heard much about it, it's a serious disease. It affects millions, primarily in Latin America. Can you give the audience the ABCs of Chagas?
Sure, I'll give it a shot. So it's as you mentioned in the intro, it's a protozoan parasite, which means it's a little bit more complex than bacteria and viruses. And that's an issue in studying it. It has complex biology. It infects virtually any mammal. So that's an advantage and a disadvantage in terms of it will never be eradicated, but it also offers options for studying it that are very powerful. It's transmitted by an insect vector. It's not easy to acquire the infection because this insect, although it inhabits houses and is a blood feeder, it doesn't pass parasites by the blood meal, but instead defecates and leaves parasites either on the skin or around the face or even on food in the house and things like that. And so transmission is not as simple as in a lot of other parasitic infections. But when the infection is acquired by humans and other animals, it is, as you mentioned, primarily, it can be acutely problematic, especially in very heavy infections. But in most humans, the infection is controlled. Oftentimes, they don't really know they're infected, and the persistence of the infection, eventually the parasite resides primarily in muscle and causes destruction of that muscle. And heart muscle in particular is the main clinical issue. So people will become infected, may not know they're infected. 10 or 20 years later, their heart is heavily damaged, and as a result of this persistent low-level infection.
So what is the problem or the difficulty with testing for this parasite to determine if treatment is successful? That's pretty much the whole purpose of your study here.
Right. So during its time in the host, it's primarily inside host cells, like muscle cells in the heart, where it replicates. And occasionally, as it replicates there, it will pop out of those cells and reinvade other cells. So unless you can, for example, biopsy muscle tissue and find those parasites that are at generally very low levels, it's hard to absolutely detect the infection. Occasionally, parasites are in the blood, and parasite DNA can be in the blood, but one has to use very sensitive methods for detecting that DNA, and that's essentially what we've tried to do in this study, is to make those methods more sensitive so that we can, even with very, very, very low level infections, we can still detect the infection.
All right, well, let's go ahead and switch to the study, and let me put it up here real quick. And it's got a long title, so bear with me. Serial deep sampling PCR of fragmented DNA reveals the wide range of Trypanosoma cruzi burden among chronically infected human, macaque, and canine hosts, and allows the accurate monitoring of parasite load following treatment. And that's published in eLife. So Dr. Tarleton, can you talk about the testing method described in the study?
Yeah, so basically what we did was to use an existing testing method that has been in use for over 20 years for detecting T. cruzi infection and Chagas disease, and that's PCR, polymerase chain reaction, where we amplify or attempt to amplify uh, parasite DNA that happens to be in the blood, either from intact parasites or even killed parasites where there's DNA still floating around there that can be amplified.
Mhm.
PCR will amplify that up to 1,000,000 fold, so something that's very rare can be actually seen then.
Right.
And so that method has been used for a long time, and we used essentially a well-worn PCR reaction. The big change we made in this study is to modify how we collect blood and how we process that blood to make that parasite DNA more accessible and detectable by PCR.
So it says serial deep sampling. So how frequently are you PCR in the tissue or the whatever source?
Right. So the main part of the study, what we did was to um, so one of the issues with T. cruzi infection, it affects mainly people in Latin America, but in the U.S., uh, there are people with T. cruzi infection. They do get infected here, but there are loads of animals that are infected with T.
Right.
cruzi, including animals that are held in captivity, that are bred for research use, like non-human primates. And so in this study, we took advantage of non-human primates in a primate facility that had become infected with T. cruzi because they live in indoor-outdoor situations. They're exposed to the vectors and they get infected spontaneously. They had been identified, and what we did was to look at a group of over 20 macaques in this case, and sampled them monthly for a year, one blood sample a month for a year. And the idea was to determine how stable their parasite load was, how detectable it was, how it varied over time, and how capable we were of really monitoring in this population the parasite burden and how it varied across the population. And the original goal was to really improve the detection level, but some of the things we determined in this study include the fact that when we looked at 20 different macaques, we saw more than 10 to the fifth, 100,000 fold difference in parasite levels in the extremes of that group of animals.
Mhm.
So there were some animals that were incredibly difficult to detect parasite DNA in, but we were able to buy serial sampling, that is, sampling monthly for a year, or even in those cases, we could also sample as frequently as a couple of times a week over a shorter period of time. But getting multiple samples was really important on
Mhm.
those cases where the parasite load was very low. And then processing that blood in a way that made sensitive detection easier. So the serial part of it is, yeah, bleeding, getting multiple bleeds of a certain volume, and in cases that was important in cases where the parasite load is very low. In cases where it's high, you only need one sample. It's quite easy. Yeah.
Yeah. So how effective was this?
Um,
Your test for cure. And
yeah. Well,
what were your
so
findings?
we studied this group of animals, and we again could see that they had a huge range of parasite loads, which um, allowed us to predict that uh, in cases where animals are treated, we would be able to tell if that treatment was highly effective or partially effective, right?
Mhm.
And so that's essentially what we did when we looked at um, some dogs that we included in the study where we had done treatment, and as well we looked at humans, not treated in this particular case, but the idea was that what we were seeing in terms of range of parasite load applied not just to macaques, but also to humans and dogs, and presumably any species that's infected by this parasite. And so, again, the bottom line was if you sample enough, that is serially sample, and the other part of the very long title is that you deep sample each one of those blood samples that you collect. And so in previous use of this sort of technology, people have taken a sample of blood, purified DNA, and then done 1 or 2 PCR reactions. And what we did was we started off, we did 10 PCR reactions. If those were negative, we did 10 or 20 or 40 or 100 more until we assured ourselves that that was clearly a negative sample. Mm-hmm. And so by doing deep sampling, we detected animals that otherwise we wouldn't have detected by just doing a simple, here's a sample of blood, do 1 PCR reaction, it's negative, we don't detect it. So
yeah,
it's a lot of work, it's expensive, but it was a very effective in detecting infection in all of these animals that we were sure had it.
So what's next?
What's next is, well, there's 2 sort of outcomes from the study. One was the unexpected one that we identified this huge, well, somewhat unexpected, that we identified this huge range in parasite load in different individuals. So all of these macaques, or all the dogs, or all the humans, they're all controlling this infection very well. But some of those animals have a 1,000,000-fold higher parasite load than other ones. And so you would presume a couple of things. One is that ones with higher parasite load might have a higher chance of developing disease more rapidly, and also that the way they're controlling that infection is different. That somebody that controls that infection really, really well, somebody that doesn't control it so well, their immune response is different. So trying to figure out now what is immunologically different in those individuals that allows that differential control is sort of a new, something that was unanticipated from the study, but has become a focus of the study. The second part was really part of the focus of the study, and that was, do we have a test that we can use when we test drugs in people or in other animals to determine if those drugs really work? Mm-hmm. And so we have started to use that in macaques who were drug treated, and shown that we can fairly easily detect rapidly those that are cured and those that are not cured. The process is still somewhat cumbersome and expensive. Doing hundreds and hundreds of PCR reactions on many individuals is not practical in a diagnostic lab. But there are new technologies out there. One of them that we've recently collaborated with and now acquired the instrumentation in the lab is from Countable Labs. And this is basically a system where one performs 30 million PCR reactions in a single tube. And so now, instead of doing hundreds and hundreds of individual tubes of PCR reactions, we do all those PCR reactions in one tube. And that cuts down on the cost and the logistic problems and things like that. So we're trying to work that process out so we can translate this useful tool for the lab into something that will be useful in a diagnostic or a drug testing clinical trial setting.
Yeah, very interesting stuff. Actually, this past week has been very interesting as far as research on Chagas. I saw there was another study concerning my home state of Florida where they're demonstrating a risk of potential local transmission here. So
yeah, there are infected vectors all over the Southeast. We're going to hear more and more about them. Again, risk to human infection in those settings is not terribly high. There's no reason to move out of the state, but uh, but it is, it is a little bothersome when you collect these insects that can transmit an infection, and in their blood meal is human blood, which means they have been feeding on humans,
Mhm.
which is what that report showed.
Yeah.
Yeah.
Any final thoughts on your study
Um,
or Chagas in
well,
general?
yeah, we're very excited about it. We took on the study because we have, in collaboration with a company, AN2 Therapeutics, we've co-developed a drug that we've shown works really, really well in the same group of non-human primates, and that is now in August going in phase 1 trials in humans, and then hopefully phase 2 sometime next year about this time, and we um, hope to be able to use this testing protocol that we've developed to really validate that drug and potentially other drugs that get developed. That's been a problem in the past, is that, unfortunately, the drugs that have been tested have all failed miserably. So it was easy to detect that they failed. But hopefully, we have a drug now that's going to succeed, and we need a way to show that they have succeeded. And this is a sort of test, I think, that will help us make that conclusion if it's the case.
Yep, yep. Sounds good. Sounds very promising. Again, the study. Let me put it up here. It's entitled "Serial Deep Sampling PCR of Fragmented DNA Reveals the Wide Range of Trypanosoma cruzi Burden Among Chronically Infected Human, Macaque, and Canine Hosts, and Allows Accurate Monitoring of Parasite Load Following Treatment." Published in eLife. And I'm going to go ahead and link to that in the show notes for people that want to check that out. It's very interesting stuff. And I want to thank you, Dr. Rick Tarleton, for sharing your time and your expertise, sir. Thank you so much.
My pleasure, Robert. Good to talk to you again.